welcome to the deep dive, everyone. Today we're taking a bit of an unexpected turn, especially for our philosopher friends out there. We're going deep into the world of molecular biology.
Oh, yeah.
You gain.
Absolutely. Always up for a good deep dive.
Perfect. So we'll be exploring this through the lens of the Stanford Encyclopedia of Philosophy's entry on molecular biology. And trust me, it's way more philosophical than you might think at first glance.
It really is. I mean, just think about it. When you start peeling back the layers of life down to genes, molecules, information, you bump right into these huge philosophical questions about reductionism, about what life even is. It's fascinating.
Totally. And what's really striking is how the history of molecular biology itself is a story of all these different disciplines smashing together. Back in the 1930s and 1940s, you had geneticists, physicists, chemists, all these brilliant minds all trying to figure out this one big mystery: inheritance. How does it work?
Yeah, and that convergence of disciplines, I think, is key to understanding why molecular biology is so philosophically rich. It's not just about one perspective. It's about all these different ways of looking at the same problem. You have someone like Thomas Hunt Morgan, right? He was all about those meticulous fruit fly experiments.
Yeah.
Linking genes to these physical structures, chromosomes.
Or chromosomes, the classic image.
Right, exactly. And that was huge because it connected this abstract idea of heredity to something you could actually see and study.
It's like suddenly those abstract laws of inheritance become real, tangible.
Absolutely. And then you have H.J. Muller, who decided to get even more hands-on. He was using X-rays to actually cause mutations in those genes. A physicist meddling with the very building blocks of life.
Wild, right. It's like, talk about a mad scientist, but in a good way, I guess. He even put out this call to action, like, who's going to step up and dive deeper? It was a challenge to the scientific community.
And guess what? People totally rose to the occasion. You had physicists like Erwin Schrödinger, the quantum mechanics genius. He wrote this book, What is Life?, where he explored the deep connections between, physics and genes.
That's so interesting. I mean, quantum mechanics and genes, what a combo.
Right. And then there was Max Delbruck, another physicist turned biologist. He used bacteriophages, those viruses that infect bacteria, to study how life actually manages to replicate itself.
So even from the very beginning, we see this inherent tension. Can we really reduce life down to its simplest parts, these molecular mechanisms? Or there's something more, something we're missing by focusing just on the molecules? I mean, that's a question philosophers have been wrestling with forever.
Absolutely. And then, boom, 1953 hits. Watson and Crick drop this bombshell, the double helix structure of DNA. It was a total game changer.
Like, whoa. The structure of life itself.
Right. And suddenly the gene wasn't just some abstract unit of inheritance. It became an informational molecule.
Informational. That's a whole new way of thinking about it.
Totally. It's like cracking a secret code. The structure of DNA gave scientists this new way to understand how it copies itself, how it actually works. And this sparked a whole new language. Information, transcription, translation. We hadn't really talked about genes like that before.
It's like biology got a software upgrade.
Yeah, exactly. And Crick, he was a big part of that. His central dogma, DNA, makes RNA. RNA makes protein. It really solidified this whole information-based view. But this raises a big question, especially for a philosopher. What does information actually mean in this context?
Right. Is it just a convenient metaphor?
Mm-hmm.
Or is there something deeper? Like, is there a fundamental link between biology and, say, information theory?
Yeah.
Could there be these universal laws of information that apply to genes and to computer programs? It's a pretty mind-bending idea.
Definitely. And just when we thought we were starting to get a handle on genes, things got even more complex.
Uh-oh, what happened?
Turns out genes are much trickier than we initially thought. They're not just these simple, straightforward stretches of DNA that each make one protein.
Wait, so it's not like a one-to-one thing?
Nope, not that simple. They can overlap. So you can have the same sequence coding for multiple proteins. They can be split up into these things called exons and introns.
Exons and introns. Okay, those are new words for me.
Yeah, think of it like this. The exons are the important bits, the coding parts, and the introns are kind of like spacers in between. And then there's this thing called alternative splicing, where the exons can get mixed and matched in all sorts of ways.
Whoa, mixed and matched, like rearranged.
Yep. And that means you can get tons of different proteins from just one gene.
It's like genes are way more versatile than we gave them credit for.
For sure. And this, of course, totally throws a wrench in that simple reductionist view of the gene. It really makes you wonder, is a gene even the right fundamental unit to be thinking about anymore? Or is there something else at play here?
So it's like we went from genes being these simple beads on a string to this whole intricate dynamic system.
Yeah.
The deeper we look, the more questions pop up.
Exactly. It's like we've only just scratched the surface.
And that's precisely where we'll pick things up next time.
Stay tuned. It's about to get even more interesting.
Can't wait.
It's really amazing, isn't it? And molecular biology just keeps throwing these curveballs at us. Like every time we think we've got a grasp on things, boom, some new discovery pops up and forces us to rethink everything.
Yeah, totally. It's like we're peeling back layers of an onion only to find, more onions underneath. But speaking of expanding horizons, this molecular revolution didn't just stay cooped up in the lab. It started branching out, influencing all sorts of different fields.
Oh, absolutely. I mean, molecular biology techniques, they became these powerful tools, you know? Suddenly, scientists could study things like how an organism develops from just a single cell, how their brains work, how their immune systems function, even how evolution plays out over time. It's wild.
It's like having this brand new lens to view the world through, right? Suddenly you're seeing details, connections, things you never even noticed before. But did this lead to any new philosophical questions? I mean, I'd imagine it had to, right?
Oh, definitely. I mean, for example, you had scientists using model organisms like, say, yeast or worms to study these really complex processes. But then it raised this question, how much can we really extrapolate from these simpler organisms to something as complicated as, a human being?
Right, that's a big question. And speaking of big, we can't forget about genomics. I mean, molecular biology went from studying individual genes to tackling entire genomes, the complete set of DNA in an organism.
The Human Genome Project, right.
Mm.
That was a huge undertaking. It was like mapping out the biological blueprint for our entire species. And it wasn't just limited to humans either. Scientists started sequencing the genomes of everything. Bacteria, plants, animals, you name it.
It was like a genomic gold rush. But with these philosophical nuggets hidden among the data. So did they find what they expected? Did it all line up neatly?
Well, not exactly. It turned out the number of genes in an organism doesn't necessarily correlate with its complexity. Like humans, for instance, we have roughly the same number of genes as a simple nematode worm.
Wait, hold on. We have the same number of genes as a worm? That's kind of humbling and mind-boggling at the same time. So what does account for all this incredible diversity and complexity we see in life, if it's not just the number of genes?
Well, this is what led to a shift in focus, right? It wasn't just about cataloging genes anymore. It became about understanding how those genes are regulated, how they're turned on and off, how they interact with each other, with the environment. It's like realizing the instructions for building a complex machine are pretty useless unless you also know the order of operations.
So instead of just a blueprint, it's more like a dynamic instruction manual, constantly being edited and rewritten based on the context. That's a pretty big shift in perspective.
Exactly. And this is where we enter the world of what's called post-genomics. And here, those huge stretches of DNA that were once dismissed as junk DNA, well, they suddenly take center stage.
Wait, so that junk DNA, it wasn't so junky after all. What's it actually doing?
Turns out it plays this crucial role in regulating gene expression. So controlling when, where, and how much of a protein is actually produced. It's like realizing those packing peanuts you were about to throw out actually contain the assembly instructions for the entire piece of furniture.
It's crazy how these seemingly insignificant parts can be so vital to the whole system. It's like molecular biology is constantly reminding us to question our assumptions, to dig deeper.
Yeah,
all right?
absolutely. And this brings us to the heart of why molecular biology is so fascinating, especially for a philosopher, I'd say. We're grappling with some fundamental questions about mechanism, about information, and even about the very definition of a gene.
Okay, color me intrigued. Let's break those down a bit. Let's start with mechanism. So we talked about how molecular biology is kind of obsessed with understanding how these intricate molecular machines operate, right? But what are the philosophical implications of that focus?
Well, for one thing, it raises questions about how complete and how useful mechanistic explanations really are in biology. I mean, can we truly understand life by just breaking it down into these super complex mechanisms? Or is there something else, something more that we're missing?
Is knowing all the gears and springs really enough to explain the beauty, the complexity of a living organism? It makes you think.
And then there's the whole question of information, right? We talked about how DNA structure led to this informational view of genes. But what is biological information really? Is it just a metaphor or is there something deeper going on there?
Yeah, we've been throwing around terms like code and transcription and translation, but what do they actually mean in a biological context? And where does this information even reside? Is it all in the DNA sequence or is it spread throughout the cell, maybe even the entire organism?
Mm.
It's a real head scratcher.
It is, for sure. And it gets even trickier when you consider the question of the gene itself. I mean, we've already seen how overlapping genes, split genes, alternative splicing, all of that stuff really challenges that simple, neat definition of a gene.
It's not just a simple one-to-one relationship anymore. It's more like this web of interactions of possibilities.
Exactly. So the question becomes, what is a gene in this complex dynamic system? Can we even define it anymore? And what does that mean for the whole idea of DNA as the sole determinant of our traits?
Like everything we thought we knew about genes is being turned upside down. It's exciting, but also kind of unsettling, you know?
That's the beauty of molecular biology, though. It forces us to constantly reevaluate what we think we know and embrace the unknown. And that's exactly what makes it such fertile ground for philosophical exploration.
I'm definitely starting to see that. But before we get too lost in the philosophical weeds, I'm curious about how scientists actually do molecular biology. What role do experiments play in all of this?
Ugh, experiments. They're not just about confirming what we already know. They're like adventures into the unknown, often leading to these unexpected discoveries that really challenge our assumptions and make us revise our theories.
Okay, I'm game for an adventure. Lead the way. Okay, so experiments. I mean, it seems like pretty much every big leap forward in molecular biology has come from some pretty ingenious experiment, right?
Yeah.
Like pushing the limits of what we even thought was possible.
Absolutely. It's not just about revealing facts. It's like experiments actually shape the way we think about biology, you know? They help us move beyond just simple correlations and actually get at the mechanisms that drive all these intricate processes we've been talking about.
So it's not just looking at cells under a microscope anymore.
No, not at all. It's about manipulating systems, isolating parts, testing ideas, and then trying to put all those pieces back together to understand the bigger picture. Remember those molecular machines we were talking about? Experiments are our way of taking them apart and figuring out like how they actually tick.
That's a great analogy. Like
Yeah.
taking apart a watch to see how all the gears and strings work together to, to keep time.
Exactly. And this experimental approach has led to some truly amazing insights, you know. For instance, the Stanford Encyclopedia mentions how back in the 1950s and 60s, scientists were really struggling to figure out the mechanism of protein synthesis. So basically, how does DNA actually instruct the creation of proteins?
I mean, that must have been a huge mystery at the time, right? Like, how does this one molecule, DNA, hold the blueprint for building all these different proteins that do, well, pretty much everything in our cells?
It was a big puzzle, for sure, and scientists tackled it from all these different angles. Some, like Watson and Crick, were working forward from DNA, you know, using techniques like X-ray crystallography, building models, trying to understand its structure, how it interacts with other molecules.
So they were starting with what they knew about DNA and then trying to work out, like, the next steps in the process.
Exactly. But then at the same time, you had these biochemists who were working backward from proteins, you know, studying chemical reactions, how the bonds form, basically trying to reverse engineer the whole process.
It's like two teams digging a tunnel from opposite sides of a mountain, hoping to meet in the middle.
That's a perfect way to put it. And that's pretty much what happened, too. They eventually converged on RNA, which turned out to be this crucial link between DNA and proteins. And this led to Watson's famous schema, DNA to RNA to protein. It became like a central dogma of molecular biology.
It's a really cool example of how experiments can really push science forward. But I imagine it's not always that straightforward, right? Like, what happens when you have multiple competing hypotheses? How do you decide which one is right?
Well, that's where this idea of a crucial experiment comes in. Ideally, you design an experiment that, you know, definitively rules out one hypothesis and supports another.
Yeah, that sounds simple enough in theory, but I bet it's a lot trickier to actually pull off.
You're telling me. The philosopher Pierre Duhem, he pointed out that to have a truly crucial experiment, you need to know, like, every possible explanation for a phenomenon, which is practically impossible.
So even if an experiment seems to confirm one idea, there's always a chance you've missed some other explanation.
Exactly. But that doesn't mean crucial experiments are useless either. Another philosopher, Marcel Weber, he argues that while we might not be able to achieve absolute certainty, we can still get pretty darn close.
How so? How can we get close to certainty if we can't rule out every single possibility?
Well, Weber analyzed a bunch of these classic experiments, like the Michelson-Stahl experiment, which confirmed the semi-conservative model of DNA replication. And he argues that while these experiments didn't rule out like every single conceivable alternative, they did eliminate the most plausible ones at the time.
So they provided like really strong evidence in favor of one explanation over the others, even if they couldn't be 100% certain.
Exactly. And this often involves what's called inference to the best explanation. So scientists aren't just blindly following the data, they're also considering which explanation makes the most sense given everything else they know about the system.
It's like being a detective, right? You have all these clues, they point you in a certain direction,
Yeah.
but you also have to use your knowledge, your intuition, to piece together the story, to build a solid case.
Perfect analogy. And in the case of the Michelson-Stahl experiment, Weber argues that, the very structure of DNA, it imposed certain constraints on how replication could possibly work.
So even though they couldn't test every single hypothetical mechanism, the ones that remained were like the most likely candidates, given what they knew about the molecular machinery involved.
Precisely. And this brings us back to something we touched on earlier. Molecular biology is really about understanding mechanisms, and experiments are the tools we use to uncover those mechanisms, to test their parts, and ultimately to understand how they generate, well, the phenomena of life itself.
So we've gone from these big, almost philosophical questions about the nature of life and information, all the way down to the nitty-gritty of how experiments are designed and carried out. It's amazing how it all connects together.
That's the beauty of molecular biology. It really bridges all these different levels of understanding, from the philosophical to the experimental.
So for all our philosopher friends out there listening, what would you say are the key takeaways? Why should they care about all this molecular biology stuff?
Well, I think, as we've seen, molecular biology is not just about memorizing a bunch of facts about cells and DNA. It throws up these really profound philosophical questions about causality, about reductionism, the nature of information, even what it means to be alive.
It's like this whole hidden world of philosophical puzzles embedded within the science itself.
Exactly. And I really believe that grappling with these questions, it can enrich our understanding of both biology and philosophy.
I totally agree. It shows how these two fields, which seem so separate on the surface, can actually inform and illuminate each other in really cool ways.
Well said. I think that's a great place to wrap up our deep dive into the fascinating world of molecular biology.
Yeah, I think so too. Thanks for joining us on this journey. It's been a real pleasure exploring these ideas with you.
Likewise, it's always a great time to dive dip with you.
Until next time.